And I went back to
it was me, that's what
like not that much. It was like because I didn't know. Oh yeah. Do you have much Sherlock?
It's better than nothing.
Yeah, and like freezing.
Yeah, it takes 10 minutes.
And I didn't even have to like take him. And he was like go home and like think about like.
Good morning. Happy Monday. Great way to start the week with cell and molecular biology. We're going to learn about the gene today. I know everyone's excited.
Oh yeah.
Learn exactly what a gene is. All right. So before we get started with class, a few announcements, just so we're on the same page about a few things. I want to let you know I will not be having office hours today. I apologize. I have to teach another class across campus. So if you have questions for me regarding the homework in particular, and you wanted to schedule some time before class on Wednesday, one of the two, just send me an email. Find a time later this afternoon or tomorrow to meet. Okay, so no office hours today. Let me know if you want to meet with me. We'll find another time. Related to the homework. So many of you noticed I made a small mistake on Friday. Posted the key rather than the questions. So, freebie for homework this week. I hope you at least attempted to look at the questions and answer them before you looked at the key. But also, I hope you noticed, I sent out a gazillion announcements. I'm sure you started ignoring them on Friday because it was Friday afternoon and you didn't want to hear from your professor. But I reposted the homework without the answers, for those of you who didn't get to it quick enough. I also put in there an extra added bonus question. Not bonus points, just an extra question that wasn't included on the key. So please be sure that you do that version of the homework. So there's an extra, if you got the one with the key, there's another question up there that you need to do that's not on the key. Okay? Question, Jessica.
When you say bonus question, it doesn't mean extra credit.
It does not mean extra points, it means extra question. Because I made a mistake, so. Bonus question is that you just get an extra question you didn't get the answer to. That's all it means. No extra points. All right, so make sure that you get that copy of the homework and that you do that extra question. All right, and you're welcome for handing out the key before I actually handed out the homework. All right, are there any questions about that? I know you are all anxiously awaiting your exam grades. You should have those done. Just to remind you, tomorrow night. Hopefully have the grades posted by Wednesday class time or sometime Wednesday anyway. I'm going to post not only the exam grades, but all of the grades up until that point, including your participation points, your quizzes, your homework. So when those get posted on Laulima, make sure that you double check them, right? Make sure that we haven't made a mistake somewhere entering the grades. Make sure that you're getting the participation points that you think you should, right? So that if you think you've been clicking, you're registered, make sure that those points are showing up. All right, so that will all happen this week. And then you will get your exams back and recitation this week. Take a look at it. Okay? Any questions related to homework, exams, etc. before we get started? All right. Then let's talk about the gene. So hopefully you are familiar with the concept of the central dogma. It describes the flow of genetic information in the cell. We've already started taking a look at the processes involved, although we didn't really specify that. We've looked at how DNA can replicate itself in detail. All right, so that's the first step. Just being able to maintain that genetic information in a high fidelity way without error. The next step is going from DNA to RNA, and that process called transcription. And that's what we're going to be taking a closer look at for the next few lectures. And then after that, we'll finish up taking a look at the process going from RNA to proteins and that process of translation. So you know what's coming up when we get to proteins. What are proteins made of? Amino acids. What do you think you're going to have to learn to draw? Amino acids. 3 amino acids. We're not there yet, but keep that in mind. All right?
Megan's like the best teacher.
Another way to put this a little more simply, DNA makes RNA, RNA makes protein. All right, what do you know about RNA? What kinds of RNA are there?
tRNA, mRNA,
I heard those two. What others? Any others? SI? SN? Any others? You know what these stand for? tRNA? Transfer RNA? Messenger RNA? Ribosomal RNA? SN? Small nuclear RNA. All right, so the 3 most commonly talked about in biology are tRNA, mRNA, and rRNA. Do all 3 of these code for proteins? Which should be a pretty straightforward question. Good, I think I got all the clicker issues sorted, so we'll get back to asking clicker questions today.
Where's the C, please?
All right, yes or no? 50-50 chance if we don't know the answer. Has everyone had a chance to answer? Everyone's still waking up? All right, I'll give you 1 more second.
When?
All right, let's take a look. What do you think? Maybe. Or not.
Do it again.
Oh, there it is. Hiding on the other screen. All right, so 50, well, not quite 50-50, but close.
How could you make it sound like that?
Anyone want to defend their answer? No one's willing to defend their answer? What does ribosomal RNA do? It makes ribosomes. All right. What does tRNA do? Transfers information. Good guess. It carries amino acids around. So what does that leave? Messenger RNA. So the only type of RNA that we have that actually codes for a protein is that messenger RNA. Okay? So even though we've got these other types of RNA and they are involved in translation, they're involved in, at least tRNA and mRNA are, they're involved in making that protein. The only one that actually codes for the protein is the mRNA. So we can change our central dogma, simplified statement. DNA makes RNA, mRNA makes protein. So we're at the step of transcription. So we're going to focus for a good portion of today, well, part of today anyway, on this idea that DNA makes RNA. So those, well, are the three main types we've talked about so far. We'll get into greater detail about rRNA and tRNA when we get to translation. mRNA is the only protein-coding RNA. When we generated our list, you also came up with snRNA, or small nuclear RNA. It turns out there are lots of different kinds of RNA. All right, and this has been one of the major discoveries in recent history. There are all these really poorly characterized, sometimes very small pieces of RNA. The only ones that are coding are those mRNAs. The rest of these are all non-coding RNA. All right? So when we're looking at transcription, we're looking at going from DNA to any of these types of RNA. So just, we'll talk about some of the small nuclear RNAs later. We'll talk about miRNA or microRNA later in the class. A lot of these we won't talk about. Small nucleolar, Peewee, interfering. So there's all sorts of different kinds. Some of which they don't even really understand what they do yet. All right, so there are lots of different types of RNA in the cell. We're going to focus mostly on those that are involved directly in that central dogma process. But I want you to know there's a large diversity out there. All right, so if DNA makes RNA, and that's the process of transcription, can you define transcription in a more specific way? Transcription is the process of well, it's so close. So you're synthesizing, so you got that right, RNA from your DNA template. So we looked at the process of DNA replication, where we're making DNA from DNA. Transcription is the process of making RNA from DNA. All right? We're taking that DNA as a template strand and producing a molecule of RNA. Unlike replication, where we are copying the whole thing, we're making a specific product. Right? So we need to know something about how the cellular machinery decides what segment of DNA to turn into RNA. So getting you started thinking about that transcription process. Another clicker question. Think about what we talked about with DNA replication and the enzymes involved. That was going from DNA to DNA. What enzyme do you think is involved in transcription, going from DNA to RNA? All right, feel free to talk to your neighbors. Right, so just like on the exam, read each of those closely. Look at all the components.
That was neat. Apparently I only read half the questions and then try to answer it. Okay, well, Ruby doesn't understand instructions at all.
Actually, like, Google came out.
You would be.
I know, also,
Yeah. It's kind of biology, doesn't it?
have you been sick yet?
We don't need that right
Then why is there E
I know, I
on
won't
there?
kiss you. I'm pretty much done. It was like a week ago. This weekend was
We're doing it in
my
Spanish? You're making it extra hard. Ah. Feel free then. Do it all in Spanish. All right, did everyone get a chance to answer?
Yes.
Whose answers in?
Whose GoPro is this?
All right, everyone get their answers in? There seems to be a few shy. All right, let's take a look and see what you think.
Okay.
All right. I think you have that figured out. That is the correct answer. All right, so we're making RNA, so it's an RNA polymerase, which is dependent on a DNA template. DNA-dependent RNA polymerase. All right, so let's get started. Before we can really talk about this process of transcription, we need to understand what the unit is that's being transcribed. So, let's talk about what a gene is for a few minutes. So get together with a few people around you to come up with a definition of a gene.
A series of base pairs.
In Spanish.
That's a team.
What's this for? You all figured out?
I don't like stupid like
I literally have like 100 dollars. I'll get an A+ for this class, probably an A+ in German. Which one is the other one? This class, I'm banking on the other side. And also don't have that. Rather that.
Hey, when
Maggie.
When do you find out? I mean, like, is it like a testing thing or do they still have to like accept it? But
All right. Everyone come up with their definition? Let's hear some. What's the definition of a gene? Hereditary information.
Portion of DNA that codes for a sequence of proteins.
Portion of DNA that codes for a sequence of a protein. Everyone's nodding their head at that one. Everyone likes that one? Any other definitions of a gene?
I would say that the gene would, the definition would have to include the fact that it actually codes for a certain type of function.
Something that codes for a certain type of function.
Kind of
A functional enzyme. It codes for a trait. It holds the trait. So the sequence contains some information. Okay? Anything else you want to add to that definition of a gene? So you're on the right track, right? So we're missing one sort of critical thing, I think. So what is the process of transcription? Simplified. DNA to RNA, not DNA to mRNA. Right? mRNA is the only one that codes for protein. So our definition of a gene has to be a little bit broader. Right? Because it codes, we're transcribing all those different kinds of RNA, not just mRNA. So certainly, there are genes, lots and lots of genes that are very important that are coding for a protein product. But if we take that word protein out of it, it's a stretch of DNA that codes for a product, codes for an RNA with some function. All right, that's not limited just to proteins because we have to make ribosomal RNAs and tRNAs and all those other types of RNA as well through the same process. So a gene is a stretch of DNA that's going to code for an RNA. All right, code for some functional cellular product. Now that we have some definition of what a gene is, we need to be able to go to the genome and identify it. What stretches are transcribed? How does the cellular machinery recognize those? All right, before we get that, how many chromosomes do humans have? Haploid number? 23. 23, right? Those is the unique set of genetic information that we have. The other 23 are copies. All right, so if we want to know how many genes we have in our genome approximately, we can multiply that number of chromosomes by how many genes we have per chromosome. See approximately how many genes we have in the human genome. Anyone have any guesses for how many genes we have approximately per chromosome? Throw out some numbers. 5,000? Anyone else want to throw out a number? 25,000? Higher? I'm going to say a million. A million genes per chromosome. Man, we have chromosome packed full of genes. Anyone else? Anyone want to do the Price is Right and go for like
1.1 million.
No? 1 cent? 1 gene per chromosome? So when we first started sequencing, we as the field of molecular biology started sequencing genomes, and it still is a big issue. How do you take a genome and predict all of the genes that it contains? All right, some of that is going to be based on what we're going to be talking about today, as far as indicators of what a gene is. But the predictions that we came up with initially were very high, and they've come down quite a bit since then. So at a very rough estimate, there are about 1,000 genes per chromosome. Which gives us approximately how many genes? 23,000 in our genome. All right, so that's what we're talking about when we're talking about genes on a chromosome. And think about how big a chromosome is. So it's not back-to-back genes. There's lots of other stuff in between those units that we're concerned about transcribing. All right, so what is a gene? We've been talking about chromosomes. Right, so there's our 2 strands of DNA, with the arrow indicating the 3' end. We can zoom in on that and look at just the region of that chromosome. Those colored lines now represent genes. Genes come in different sizes and shapes. Nice diversity. What is the first thing you would notice about genes just based on this picture? And it's the way that it has to be because we know that it transcribes a finite product. They have a beginning and an end, right? There's some way that there's something that defines the start and end of that gene. Right? They have some sort of limit. So we can define it as a discrete section of the chromosome. If we zoom in on just a single gene, we can look at within those limits, what's going on? What are the main components that we're interested in? All right, so we have our limits there on the end. What we're calling a gene is that entire part in yellow. What is contained within that part of the chromosome? Good. I have that as step 2. What's step 1? So there must be a region converted to RNA. So some portion of that gene is the part that's going to get transcribed into RNA. Notice it's not the entire component of that gene, because there has to be the instructions. This is where you start, this is where you end, contained within that unit. Sorry, say that again. What's the question? We're talking about eukaryotic, yes. We haven't gotten to introns and exons. This is just one unit that's transcribed. We're building up to that. All right, so we have a transcribed region, also called the transcription unit, or segment of DNA that generates a single continuous RNA molecule. All right, again, notice that is not that entire stretch of gene, what we're calling a gene. All right, so let's think a bit about this transcription unit. Remember that we're going from DNA to RNA. We have 2 strands. So how many possible transcripts can be made from this transcription unit? The chunk of chromosome, we've defined the transcription unit, so it generates one continuous piece of RNA. How many possible transcripts can be made from this? All of the polymerases work the same way. It's in the difference is the template versus what they're making. Same general principle of how to make it.
Because it didn't even know. Where did she go? I just said. I was like, wait, I thought that was 23. Yeah.
She's like,
No, she was just funny. And she was actually in my classroom. There's a thing called this classes. That was the question on me.
How many unique transcripts? Let me define it that way.
What?
Okay. Are people answering and my clicker is not recording, or are you guys still?
Everyone's confused.
Everyone's confused.
You should tell us the answer. It'll be a preview of the day.
I'm sorry, I should tell you the answer?
Yeah, it'll be the preview of the day.
All right, I'll give you a hint. How about that?
Yes.
So we're talking about unique transcripts generated by an RNA polymerase. What does RNA polymerase need to work? The DNA template. How many DNA templates are there?
2.
But how many unique transcripts can be made from that region of chromosome?
Wait, which is
No, it's not going through. It's not going through.
All right, everyone get their answers in?
No. There's only 23.
It's not changing.
Is it frozen? Let's try this again.
I do.
Reopen it. All right, try now.
There we go.
All right, everyone get their answers in?
No.
Frozen again? All right. I will keep working on this. Apparently only the first 20 of you can answer today. So let's do this. Hands for 1 unique transcript, hands for 2 unique transcripts, hands for 3 unique transcripts, hands for 4 unique transcripts. Anyone says can't tell, unknown. All right. I think my hint helps. You can generate 2 unique transcripts from this region of chromosome. Remember, you have 2 templates. They're complementary, but they're anti-parallel, meaning you're going to synthesize them in opposite directions. Therefore, you can make 2 unique transcripts, one off of each strand of DNA. Does everyone see that? All right, so if we can make 2 possible transcripts through the process of transcription from any stretch of a chromosome, how do we figure out which one is made? So how many transcripts are actually made from a gene? And for 1? And for 2. All right, so to maintain the integrity of whatever product you're coding for, you want to make sure you only transcribe one of those templates. Otherwise, you're making 2 different products. All right, so in general, the process of transcription is only using one of those strands as a template. All right? What about this question? Trying to get this out of the way. All right. Which transcript is made? We know it can make 2. Possibly it can make 2 different transcripts. We know that only one is actually made. Which strand is used as a template? See if it works for this question.
Yeah, but there's no. No.
Can you tell? Have I given you enough information to tell? All right. You can't tell yet. You don't have enough pieces of the gene put together. All right. We're going to stop this. All right. I haven't given you enough information to tell which template is actually being transcribed. What piece of information is going to tell us that? What piece of the gene don't we have represented yet? The promoter. All right, so the promoter controls which transcript is made. All right, so we answered this first in lecture. You guys like to jump ahead. All right, so what does it do? What's the specific job of the promoter? So it indicates where to start transcription? How does it do that? It does involve a specific set of proteins. All right. How do we know that it's, we know that it's determining the direction of transcription. What happens at the promoter site? I heard it.
RNA polymerase binds.
It binds RNA polymerase. So it's the recognition sequence for that enzyme that's going to be making our RNA transcript. So the purpose of the promoter is to bind our RNA polymerase. There it is. In case you had any doubt. RNA polymerase. All right? How do we know, how does RNA polymerase know to bind in that direction instead of that direction? What about the promoter? So if we're only generating template, or we're only generating transcript from one strand, that means that that can't happen. You can't have the polymerase bind in either orientation. There has to be a specific orientation, which suggests what about the promoter? It has directionality. All All right, we've talked about a lot of things that have directionality so far. Nucleic acids, nucleotides, DNA strands. Promoters also have directionality to ensure that the promoter sits down at the right place to start transcription at the right place and in the right direction. All right, so we know that it has directionality. So that polymerase only binds in one orientation. Not like this. All right, that little blue squiggly thing is the transcript being generated in my notation. Looks like a worm. All right, so we know that it must have directionality, which means that we have to add that piece of information to our drawing of the gene. How would you add directionality to your drawing of a promoter? An arrow would work. Shading would work. Something to indicate that the promoter is generating a transcript in a specific direction. So that the polymerase can only bind in that direction. All right, so polymerase, we're talking about a polymerase, which means it works just like a DNA polymerase did that we talked about in replication. Right, the difference being you're going from DNA to RNA rather than DNA to DNA, but the process is the same. What direction is RNA going to be synthesized in? 5' to 3'. Always happens the same way. So let's play a game the last few minutes of class. The promoter game. So I'm going to give you a drawing of a gene that contains the elements we've talked about so far. That transcription unit, the promoter, as I've shown you with directionality. And you have to tell me which strand is the template. By giving you that promoter, actually all I'm showing you is the promoter, I think. There's enough information there to figure out which is the template strand. Remember how it's being synthesized and what that means about the template. All right, top or bottom strand. If that's our promoter. Discuss for a minute and then I'll have you raise your hand. Look at it carefully. Which direction is it being transcribed in? Which means what about the template? The squiggly line is the blue wormy squiggly line is the transcript being made.
Oh wait, yeah. Well, I mean, because the blue thing. I was thinking we didn't have to follow the blue thing.
Talk it over.
You have to take a picture.
I don't know, she always posts late.
Turns into like a.
Wait, but why does it say upstream?
I think it's like a giraffe. See, she's
Oh
always
yeah, okay.
All right, does everyone have it figured out? All right, by show of hands, which strand is the template? Top? Bottom?
What's up?
Defend your answer. Top. Why do you think it's the top? No one's going to defend their answer for the top? Bottom? People?
It goes 5' to 3' and And then this is the exact thing that you showed us in the previous picture.
Why is it the bottom? If we're generating a 5' to 3' to the right side of the board, oh, I actually have this. So 5' to 3', what orientation must the template be in? 3' to 5'. It has to be the opposite oriented template. Remember when we talked about DNA replication? The template is oriented 3 to 5. It's the new strand is synthesized 5 to 3. All right, so if I give you a chromosome or a piece of DNA like this and give you promoter orientation, you should be able to figure out what the template strand is. So what about this one? Discuss this for a couple of minutes. I haven't given you the transcript. Figure out which one is the template strand. What direction is it being transcribed in? What does that mean about which one must be the template?
There's no way to like I would assume it would go that way because it's closer to like this side, so it would start from the first side.
The promoter is going to bind the polymerase so that it moves to the right. Well, the way that I've drawn it, from green to white.
The green?
Yeah.
With the green?
Yeah.
I think it has to be.
I will indicate in some obvious way which way the promoter is on the exam.
You know, like from Hercules?
Alright, so that, okay, got that the right way? That's our gene, or our promoter anyway. Which is the template strand? Hands for A, hands for B. Alright, let's look at this for a minute. So if this is our promoter, it's going to bind the RNA polymerase so it moves that way. Generates things from 5' to 3'. Which is the template? Which it has the opposite orientation? Top strand. Correct. Alright, everyone see that? So I don't need to draw that transcript in for you if I give you the promoter and show you which way it's going to bind RNA polymerase. Alright? So we talked today about the limits. We talked about the transcribed region, that transcriptional unit. And we talked about the promoter. And with those three pieces of the gene, you can figure out which strand, actually with just the promoter, you can figure out which strand is the template for transcription. Alright? So we're going to end there. We'll pick up on Wednesday, continuing to talk about the components of the gene. We still have a few to go. Your homework we do on Wednesday. Don't forget that extra bonus question. Both questions that you just one you don't have the answer to. No extra points.
I don't even know what.
1 o'clock, please.